So the next thing we want to be able to understand is how we get the information out of DNA and do stuff with it. And before we can talk about that process, which is grouped into two sub-processes called transcription and translation, in order to really understand those, we have to understand something about the other nucleic acid that's a player in all of this, called ribonucleic acid, or RNA. Now, you've heard of RNA, and many of you know what RNA is and what it does, but we're going to talk a little bit about how RNA is similar to DNA, how it's different, and essentially what it's used for. To start with, this little diagram is something that we call the central dogma of molecular biology. And for a long, long, long time, it has been the basic idea behind how information comes out of the DNA and what it's used for. So at the very top here we have DNA, which is the information molecule. DNA can copy itself, copy that information, and it can give it to other cells. But if we want to use that information, we need to use RNA. And so what happens is the information in DNA is copied into RNA. That's called transcription. That RNA goes outside of the nucleus into the cytoplasm of the cell. It meets up with something called a ribosome, and it is translated, much the same way that you would translate something into a foreign language. The ribosome translates the information from the language of nucleic acids into the language of proteins, and a protein is made. And essentially, that's all there is to this idea of transcription and translation. Take the information out of DNA, copy it into a slightly different form, RNA, take that message out into the cytoplasm, meet a translator, the ribosome, and make a protein. And then proteins, we know, do things in the cell. They are the enzymes. They do all the chemistry. So obviously, if something's wrong with the message, the information inside the DNA, then something's going to be wrong with the protein, and you're going to have some problems. Maybe something's wrong with the RNA that's produced. You're going to have something wrong with your protein, and you're going to have some problems. So that's the central dogma of molecular biology. RNA is the stepping stone between the language of genes, nucleic acids, and the language of proteins. Okay? So what is this RNA stuff? Well, a couple of things about ribonucleic acid. It's very similar to DNA, but it has a few very important exceptions. The first is that RNA is a single-stranded molecule instead of DNA being double-stranded. So RNA simply has a sugar phosphate backbone with these bases sticking out, kind of like a comb. Instead of deoxyribose as the sugar, we use ribose. Remember, way back at the beginning of the semester, we learned that ribose and deoxyribose are almost identical in structure, except for the missing oxygen at the 2' carbon. So ribose has that oxygen there, but ultimately it's chemically very similar to deoxyribose. And the third and probably most important difference here is instead of thymine, RNA uses uracil. And uracil is very similar to thymine in structure. It is a single ring. Okay, so it's still a pyrimidine, but it's got slightly different functional groups on it, and that gives it a little bit of a different set of properties. There are different types of RNA for different functions in your cell, and they all have a slightly different conformation or shape. Now we're going to only talk about 3 of them here, but there are many, many others that you'll maybe hear about over the course of your reading or research. There are many microbiologists that would conjecture that RNA is the most important molecule in life, because while DNA carries the information, without RNA, that information does nothing. It sits there, and it never gets used. So here's a little drawing of the difference between the two. Over here on the left we have RNA, and on the right we have DNA. And you can see that RNA is single-stranded. It still coils into a helix, but it's only single-stranded. 3 of the 4 bases are exactly the same, cytosine, guanine, and adenine, but thymine has been replaced with this uracil, and if you look at it carefully, you'll see that the major difference between thymine and uracil is that in thymine there is a methyl group, and in uracil there isn't. But other than that, it should chemically behave very, very similar to thymine, and it does. There are 3 types of RNA that we need to know about right now in order to understand transcription and translation. First is called messenger RNA, or mRNA. Sometimes this is also called a transcript. When you copy something, you get a transcript. This is the copied piece of information from the genes. We have ribosomal RNA, which is called rRNA. You see in that name the word ribosome. Well, ribosomal RNA is used to make up ribosomes. And we have transfer RNA, or tRNA. And we'll talk a little bit about that in a second. So let's start with messenger RNA, also called a transcript. This is the one that carries the information from the DNA into the cytoplasm of the cell. So messenger RNA is the one that goes from the nucleus out into the cytoplasm. DNA doesn't do that. DNA doesn't get out of the nucleus, but messenger RNA is how we get the information out. Messenger RNA is like a sentence divided into 3-letter words. Okay, so think of it as a long sentence. It's divided into 3-letter words. Each word is a codon. It's called a codon. Okay, it's made of 3 bases. So A, G, C, T, et cetera, et cetera. Each 3 letters is one of the words, and it's called a codon. Each codon, each word, specifies a certain amino acid to be placed into a protein. So that means that the number and sequence of those bases in their RNA specifies a specific number and sequence of amino acids that make up the proteins. That's how it works. Because genes are different lengths, the messenger RNA strands that you'd find in a cell are different lengths because they're copied from the gene. And depending on what the cell does, it's going to have maybe lots of messenger RNA strands or maybe only a few. If you have a cell that produces a lot of different kinds of proteins, like a liver cell, lots of different types of proteins, enzymes, lots of messenger RNA transcripts. If you have a cell like a skin cell that maybe makes a few proteins, you may maybe only have a few messenger RNA transcripts. So it depends on what the cell is doing with them. Ribosomal RNA. Now, a ribosome, which is where protein is made mostly of RNA. It's made of the stuff that it ends up looking for to work with. Well, that would make sense, right? It'd be easier for RNA to find itself in the cell. But there are some proteins involved as well. The ribosome is where protein synthesis occurs. It has 2 pieces, 2 subunits that come together when protein synthesis is going to happen. So they're separate normally. They attach to one another when they're ready to do their job. There's another function of ribosomal RNA that's kind of interesting, forming something called ribozymes. Now you'll notice that Z-Y-M-E at the end sounds like enzyme, right? And an enzyme is what kind of molecule? It's a protein. But a ribozyme is actually made of ribosomal RNA. And it acts like an enzyme. Its specific job is it hooks these amino acid pieces together to make proteins. Polypeptides, we call them, until they fold up. So this is cartoon drawing. This is actually right from your book. It's a cartoon drawing of what a ribosome might look like. Large subunit, small subunit, kind of looks like a duck. As a matter of fact, that's why that little projection is called a bill. Okay? And it's not a smooth thing like this, though. Remember, it's made of it's a molecule. It's just a giant molecule. So it doesn't have a smooth surface. It's all these little atoms and their electrons and such kind of hanging around, packed in closely into a 3-dimensional form. But that's kind of what it looks like. Finally, we've got transfer RNA, or tRNA. And this is a connector molecule. tRNA is, on one end of it, it's attached to an amino acid. And on the other end, it can attach to, or it can be attracted to, a messenger RNA transcript. The nature of tRNA is it's very small. It's only about 75 to 80 bases in length. So it's a tiny little molecule. And because it's single-stranded and those bases are sort of sticking out, it can actually fold. And transfer RNA molecules fold into sort of a cloverleaf shape. I'll show you a picture of it in a second. On one of the loops of the cloverleaf, there are 3 bases, 3 nitrogenous bases, 3 letters. And those are very important because they are specifically ordered to be complementary to one of the codons in the messenger RNA that this transfer RNA is going to go look for. So those 3 bases on the transfer RNA are called an anticodon, and they complement or match up with a specific set of bases on a messenger RNA strand. And because the transfer RNA is attached to an amino acid at the other end, every time those 3 letters occur in the messenger RNA, the transfer RNA will bring in that specific amino acid. Okay? So transfer RNAs are amino acid specific, meaning whatever the anticodon says in that one loop on the transfer RNA, it's always going to have the same amino acid attached to its other end. That's how you can control which amino acids get into your protein, because only certain transfer RNAs will come in to the messenger. Here's an example. So these are just 2 cartoon drawings of a couple of transfer RNA molecules. The one on the left has the amino acid serine. That's the red block with the SER in it. That's representing the amino acid serine. It's attached to that end. The other end of the molecule, the loop that's sort of pointing towards the bottom, has 3 bases: uracil, cytosine, and adenine, UCA. Anytime you have a tRNA molecule with UCA in that position, it's always going to have a serine attached to the end of it. When it encounters a messenger RNA strand, that's the purple line at the bottom, when it sees AGU, AGU is complementary to UCA. Anytime there's an AGU in the transcript, well, that means that that transfer RNA will come in and it'll bring its serine with it. Similarly, anytime there's a UAC in the transcript, it will attract a transfer RNA that has AUG in the anticodon position, and that one will have a tyrosine attached to it. So the ribosome does all of this sort of holding and things in position, but they'll match up on their own, and it'll bring in a certain sequence of amino acids to make a certain protein. Here's a more detailed version, and the only point of this particular picture is so that you can see how this thing folds into a cloverleaf. You'll notice that there are, on each of the arms of the cloverleaf, there are places where there are lines drawn. Well, those are hydrogen bonds. G's with C's, and occasionally you'll see a weird thing that doesn't exactly match up, but G's with C's form 3 hydrogen bonds, and that can hold this molecule in its shape. If you look down at the bottom, you'll see there's the anticodon, and it's matched up with a messenger RNA codon. There's more going on here in this picture, but that's all you really need to know about it. So to summarize, 3 types of RNA: messenger RNA, somewhere between 500 and 3,000 or more nucleotides in a row, and that is the encoding of the amino acid sequence. It's the information from the gene. rRNA, ribosomal RNA, 100 to 3,000 nucleotides in length. That's what coils up and associates with proteins to form ribosomes, which catalyze or hold on to and allow for an easier protein synthesis. And transfer RNA, 75 to 80 nucleotides in length, very short, folds up into a cloverleaf shape. That's the molecule that holds on to the amino acid on one end and is attracted to a specific series of 3 bases in the messenger RNA to bring in that particular amino acid. Okay? A lot of information in this video. Go back, watch it again, take good notes, fill out your summary, and we'll see you next time.